Chapter 19 Excretory Products and Elimination. 1. 3. Major forms of nitrogenous waste are ammonia, uric acid, urea. 2. Ammonia, uric acid, urea. 3. Ammonia, uric acid, urea. 2. Most toxic nitrogenous waste is ammonia, ammonia, ammonia. 3. Least toxic nitrogenous waste is uric acid, uric acid, uric acid. 4. Ammoniteric X. Bony fishes, aquatic amphibians and aquatic insects. Bony fishes, aquatic amphibians and aquatic insects. Bony fishes, aquatic amphibians and aquatic insects. 5. Ammonia is excreted by dash across the body surface or through dash in fishes, diffusion, gills, diffusion, gills, diffusion, gills. 6. Ammonia is excreted as NH3. F. As ammonium ion NH4*. F. As ammonium ion NH4*. F. As ammonium ion NH4*. 7. Kidney do not play a significant role in the removal of ammonia. T. 8. Uretelic X. Amphibians, marine fishes, mammals. Amphibians, marine fishes, mammals. Amphibians, marine fishes, mammals. 9. Ammonia produced is converted into urea in the dash of these animals and released into the blood, which is excreted out by the dash, liver, kidney, liver, kidney, liver, kidney. 10. Urea is retained in the kidney matrix of some animals because, to maintain desired osmolarity, to maintain desired osmolarity, to maintain desired osmolarity. 11. Uricotelic X. Eel, reptiles, birds, land snails and insects. Eel, reptiles, birds, land snails and insects. Eel, reptiles, birds, land snails and insects. 12. Uric acid is secreted in the form of dash or dash, pellets or paste, pellets or paste, pellets or paste. 13. Proteinephridia, also called dash, are excretory structures of flamed cells, X, platyhelminthes, rotifers, some annelids and cephalochordata, i.e. Amphioxus. Flamed cells, X, platyhelminthes, rotifers, some annelids and cephalochordata, i.e. Amphioxus. Flamed cells, X, platyhelminthes, rotifers, some annelids and cephalochordata, i.e. Amphioxus. 14. Protoniuridia are primarily concerned with nitrogenous waste removal, F primarily concerned with osmoregulation, F primarily concerned with osmoregulation, F primarily concerned with osmoregulation. 15. Nephridia are found in annelids, annelids, annelids. 16. Excretory structure of most insects as Malpighian tubules, Malpighian tubules, Malpighian tubules. 17. Malpighian tubules help only in the removal of nitrogenous wastes, not osmoregulation. F. Both. F. Both. F. Both. 18. Antennal glands, also called green glands, green glands, green glands. 19. Antennal glands are present in dash like dash, crustaceans like prawns, crustaceans like prawns, crustaceans like prawns. 20. Human urinary system is shown in diagram. I. Label A to L. Diagram. I. A. Adrenal gland. B. Renal artery. C. Renal vein. D. Kidney. A. Dorsal aorta. F. Ureter. G. Urinary bladder. H. Urethra. I. Cortex. J. Medulla. K. Pelvis. L. Inferior vena cava. Diagram. I. A. Adrenal gland. B. Renal artery. C. Renal vein. D. Kidney. A. Dorsal aorta. F. Ureter. G. Urinary bladder. H. Urethra. I. Cortex. J. Medulla. K. Pelvis. L. Inferior vena cava. Diagram. I. A. Adrenal gland. B. Renal artery. C. Renal vein. D. Kidney. A. Dorsal aorta. F. Ureter. G. Urinary bladder. H. Urethra. I. Cortex. J. Medulla. K. Pelvis. L. Inferior vena cava. 21. A longitudinal section of the kidney is shown in diagram. 2. Label A to L. Diagram. 2. A. Medullary pyramid. B. Renal column. C. Cortex. D. Renal capsule. A. Ureter. F. Renal pelvis. G. Renal vein. H. Renal artery. I. Calyx. Diagram. 2. A. Medullary pyramid. B. Renal column. C. Cortex. D. Renal capsule. A. Ureter. F. Renal pelvis. G. Renal vein. H. Renal artery. I. Calyx. Diagram. 2. A. Medullary pyramid. B. Renal column. C. Cortex. D. Renal capsule. A. Ureter. F. Renal pelvis. G. Renal vein. H. Renal artery. I. Calyx. 22. Kidneys are dash in color. Reddish brown, reddish brown, reddish brown. 23. They are situated between dash and dash vertebrae. Last thoracic and third lumbar. Last thoracic and third lumbar. Last thoracic and third lumbar. 24. The kidney is located close to the dorsal, ventral, inner, outer wall of the abdominal cavity. Dorsally, inner. Dorsally, inner. Dorsally, inner. 25. Kidney dimensions, length, width, and thickness are 10-12 cm length, 5-7 cm width, 2-3 cm thickness. 10-12 cm length, 5-7 cm width, 2-3 cm thickness. 10-12 cm length, 5-7 cm width, 2-3 cm thickness. 26. Average weight is 120 to 170 g. 120 to 170 g. 120-170 grams. 27. Ureter, blood vessels, and nerves enter through hilum. 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 28. Towards the center of the kidney's inner, outer, concave, convex surface is a notch called hilum. Inner, concave, inner, concave, inner, concave. 29. Funnel-shaped space is called dash with projections called dash, renal pelvis, calyces. Renal pelvis, calyces. Renal pelvis, calyces. 30. Renal pelvis is present inner to hilum. T. 31. Outer layer of the kidney is a tough capsule. T. 32. Kidney is divided into two zones, namely outer cortex and inner medulla. Outer cortex and inner medulla. Outer cortex and inner medulla. 33. Cortex extends in between medullary pyramids as renal columns called renal columns of Bertini. Renal columns of Bertini. Renal columns of Bertini. 34. Medullary pyramids project into the calyces. 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 35. Total No of nephrons in the human body is 2 million. 2 million. 2 million. 36. A diagrammatic representation of a nephron is shown in diagram 3. Label A to K. Diagram 3. Nephron A, efferent arteriole, B. Afferent arteriole, C. Glomerulus, D. Bowman's capsule, A. Descending limb of loop of Henle. F. Ascending limb of loop of Henle, G. Henle's loop. H. Vasa recta, 1 collecting duct, J. Distal convoluted tubule, K. Proximal convoluted tubule. Diagram 3. Nephron A, efferent arteriole, B. Afferent arteriole, C. Glomerulus, D. Bowman's capsule, A. Descending limb of loop of Henle. F. Ascending limb of loop of Henle, G. Henle's loop. H. Vasa recta, 1 collecting duct, J. Distal convoluted tubule, K. Proximal convoluted tubule. Diagram 3. Nephron A, efferent arteriole, B. Afferent arteriole, C. Glomerulus, D. Bowman's capsule, A. Descending limb of loop of Henle. F. Ascending limb of loop of Henle, G. Henle's loop. H. Vasa recta, 1 collecting duct, J. Distal convoluted tubule, K. Proximal convoluted tubule. 37. Nephron 2 parts are glomerulus and renal tubule. Glomerulus and renal tubule. Glomerulus and renal tubule. 38. Glomerulus is a tuft of capillaries formed by the afferent arteriole. Afferent. 39. Blood from the glomerulus is carried away by the efferent vein. F. Efferent artery, F. Efferent artery, F. Efferent artery. 40. Bowman's capsule is a part of the renal tubule. T. 41. Cup-like structure is Bowman's capsule. Bowman's capsule. Bowman's capsule. 42. Dash encloses the glomerulus. Bowman's capsule. Bowman's capsule. Bowman's capsule. 43. Identify the structure shown in diagram 4. Label A, B, C and D. Diagram 4. Malpighian body, renal corpuscle. A. Afferent arteriole, B. Efferent arteriole, C. Bowman's capsule, D. Proximal convoluted tubule. Diagram 4. Malpighian body, renal corpuscle. A. Afferent arteriole, B. Efferent arteriole, C. Bowman's capsule, D. Proximal convoluted tubule. Diagram 4. Malpighian body, renal corpuscle. A. Afferent arteriole, B. Efferent arteriole, C. Bowman's capsule, D. Proximal convoluted tubule. 44. Glomerulus plus Bowman's capsule equals Malpighian body, renal corpuscle. Malpighian body, renal corpuscle. Malpighian body, renal corpuscle. 45. PCT is highly coiled. T. 46. Dash is hairpin shaped. Loop of Henle. Loop of Henle. Loop of Henle. 47. Collecting duct opens into renal pelvis, renal pelvis, renal pelvis. 48. Only the component of the nephron which dips into the medulla is. Loop of Henle. Loop of Henle. Loop of Henle. 49. Name the two types of nephrons. Cortical and juxtamedullary. Cortical and juxtamedullary. Cortical and juxtamedullary. 50. Loop of Henle is long in cortical nephrons. F. 51. Juxtamedullary nephrons are present in more northern cortical nephrons. F. Vice versa is true. F. Vice versa is true. F. Vice versa is true. 52. Vasa recta is absent or highly reduced in cortical nephrons. T. 53. The three processes for urine formation are glomerular filtration, reabsorption, and secretion. Glomerular filtration, reabsorption, and secretion. Glomerular filtration, reabsorption, and secretion. 54. On average, dash milliliter of blood is filtered by the kidney per minute. 1100-1200 milliliter, 1/5 of blood pumped by heart. 1100-1200 milliliter, 1/5 of blood pumped by heart. 1100-1200 milliliter, 1/5 of blood pumped by heart. 55. Filtration occurs through three layers. Name them. Endothelium of glomerular blood vessel, basement membrane, epithelium of Bowman's capsule. Endothelium of glomerular blood vessel, basement membrane, epithelium of Bowman's capsule. Endothelium of glomerular blood vessel, basement membrane, epithelium of Bowman's capsule. 56. The epithelial cells of Bowman's capsule are called podocytes. 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 57. Minute spaces between them are called dash or dash. Filtration slits or slit pause. Filtration slits or slit pause. Filtration slits or slit pause. 58. Protein also passes onto the lumen of Bowman's capsule. F. 59. Glomerular filtration is called ultrafiltration because all constituents except protein pass to the lumen. All constituents except protein pass to the lumen. All constituents except protein pass to the lumen. 60. Normal GFR values are dash milliliter per minute, i.e. dash lit per day but urine released in a day. 125 milliliters per minute of 180 lit per day, 1.5 lit per day. 125 milliliters per minute of 180 lit per day, 1.5 lit per day. 125 milliliters per minute of 180 lit per day, 1.5 lit per day. 61. JGA full form. Juxtaglomerular apparatus. Juxtaglomerular apparatus. Juxtaglomerular apparatus. 62. JGA is formed by the cellular modification of DCT and afferent arteriole. DCT and afferent arteriole. DCT and afferent arteriole. 63. A fall in GFR activates the dash cells to release dash. JG, renin, JG, renin, JG, renin. 64. Nearly _% of filtrate has to be reabsorbed by renal tubules. 99. 65. Substances reabsorbed actively are glucose, amino acid, and Na+. Glucose, amino acid, and Na+. Glucose, amino acid, and Na+. 66. Substances reabsorbed passively are nitrogenous waste. Nitrogenous waste. Nitrogenous waste. 67. Reabsorption of water also occurs actively in the initial segments of the nephron, F passively, F passively, F passively. 68. Tubular cells secrete which three things into the filtrate? What does it help to maintain? H+, K+, and ammonia, ionic and acid-base balance. H+, K+, and ammonia, ionic and acid-base balance. H+, K+, and ammonia, ionic and acid-base balance. 69. PCT is lined by dash epithelium. Simple cuboidal brush border. Simple cuboidal brush border. Simple cuboidal brush border. 70 to 80% of dash and dash are reabsorbed in PCT. Electrolyte in water. Electrolyte in water. Electrolyte in water. 71. PCT helps to maintain dash and dash of the body fluids. pH and ionic balance, pH and ionic balance, pH and ionic balance. 72. PCT secretes dash into the filtrate and absorbs dash from it. H+, K+, and ammonia, absorb, HCO3. H+, K+, and ammonia, absorb, HCO3. H+, K+, and ammonia, absorb, HCO3. 73. Reabsorption is minimum in the ascending, descending limb. Ascending limb. Ascending limb. Ascending limb. 74. What plays a significant role in the maintenance of the high osmolarity of the medullary interstitium? Loop of Henle. Loop of Henle. Loop of Henle. 75. Descending limb is permeable to dash and impermeable to dash. Water, electrolytes. Water, electrolytes. Water, electrolytes. 76. Ascending limb is impermeable to dash but permeable to dash. Water, electrolytes. Water, electrolytes. Water, electrolytes. 77. Ions are only transported passively in the ascending limb. F, actively to. F, actively to. F, actively to. 78. Conditional reabsorption of dash and dash takes place in DCT. Water and Na plus. Water and Na plus. Water and Na plus. 79. DCT also reabsorbs dash and selective secretion of dash also occur. HC03, selective secretion of hydrogen, potassium, and NH3. HC03, selective secretion of hydrogen, potassium, and NH3. HC03, selective secretion of hydrogen, potassium, and NH3. 80. A large amount of water can be reabsorbed from collecting duct, collecting duct, collecting duct, collecting duct. 81. Collecting duct allows the passage of urea into the medullary interstitium. T. 82. Collecting duct causes selective reabsorption of H plus and K plus. F. Selective secretion of H plus and K plus. F. Selective secretion of H plus and K plus. F. Selective secretion of H plus and K plus. 83. Dash and dash play a significant role in concentrating urine. Loop of Henle and vasa recta, loop of Henle and vasa recta, loop of Henle and vasa recta. 84. The countercurrent mechanism operates between the vasa recta and Henle's loop. F. 85. The countercurrent is formed between the two limbs of Henle's loop. T. 86. The countercurrent is formed between the two limbs of the vasa recta. T. 87. Osmolarity gradient from the cortex to inner medullary interstitium is dash mosmol 1. 300, 1 200. 300, 1 200. 300, 1 200. 88. This gradient is mainly caused by nickel and urea. Nickel and urea. Nickel and urea. 89. Small amounts of urea enter the thin, thick segment of the ascending, descending limb of Henle's loop, which is transported back to the interstitium by DASH, thin, ascending, collecting duct, thin, ascending, collecting duct, thin, ascending, collecting duct. 90. Nickel is returned to the interstitium by ascending portion of the vasa recta. Ascending portion of the vasa recta. Ascending portion of the vasa recta. 91. Human kidneys can produce urine nearly dash times concentrated than the initial filtrate formed. 4. 92. Kidney function is regulated by hypothalamus, JGA, and heart. Hypothalamus, JGA, and heart. Hypothalamus, JGA, and heart. 93. Osmoreceptors are activated by changes in blood volume, ionic concentration, and excessive loss of body fluid. Changes in blood volume, ionic concentration, and excessive loss of body fluid. Changes in blood volume, ionic concentration, and excessive loss of body fluid. 94. AD also called vasopressin, vasopressin, vasopressin. 95. AD prevents dash. F. Water loss through urine. F. Water loss through urine. F. Water loss through urine. 96. ADH also has a constrictor effect on blood vessels, thus increasing blood pressure. T. That's why it is called vasopressin, vasovessel, pressin, constrictor. T. That's why it is called vasopressin, vasovessel, pressin, constrictor. T. That's why it is called vasopressin, vasovessel, pressin, constrictor. 97. JG cells are activated by dash and release dash. Fall in GFR, renin, fall in GFR, renin. Fall in GFR, renin. Fall in GFR, renin. 98. Renin converts dash to dash. Angiotensinogen to angiotensin. Angiotensinogen to angiotensin. Angiotensinogen to angiotensin. 99. Angiotensin I is converted to angiotensin 2 by dash enzyme found in dash. ACE angiotensin convertase enzyme, lungs. ACE angiotensin convertase enzyme, lungs. ACE angiotensin convertase enzyme, lungs. 100. Angiotensin 2 functions powerful vasoconstrictor, activates adrenal cortex to release aldosterone, powerful vasoconstrictor, activates adrenal cortex to release aldosterone, powerful vasoconstrictor, activates adrenal cortex to release aldosterone. 101. Aldosterone causes reabsorption dash and dash from the proximal, distal parts of the tubule. Water and Na plus, distal. Water and Na plus, distal. Water and Na plus, distal. 102. ENF decreases blood pressure by causing diuresis. F. By vasodilation. F. By vasodilation. F. By vasodilation. 103. ENF causes vasodilation, vasodilation, vasodilation. 104. Dash mechanism checks the RAS mechanism. ENF, ENF, ENF. 105. What is the first event that initiates micturition? Stretching of the urinary bladder, stretching of the urinary bladder, stretching of the urinary bladder. 106. CNS initiates the contraction of dash and relaxation of dash during micturition. Smooth muscles of the bladder, internal urethral sphincter, smooth muscles of the bladder, internal urethral sphincter, smooth muscles of the bladder, internal urethral sphincter. 107. An adult human excretes dash liters of urine per day. 1 to 1.5. 1 to 1.5. 1 to 1.5. 108. The urine pH is dash. F, light yellow. F, light yellow. F, light yellow. 109. Urine is white in color. F, F, F. 110. Dash gm of urea is excreted out per day. 25 to 30 gm. 25 to 30 gm. 25 to 30 gm. 111. Presence of glucose in urine is called dash and ketone is called dash. Glycosuria, ketonuria. Glycosuria, ketonuria. Glycosuria, ketonuria. 112. Both these conditions are indicative of dash, diabetes mellitus, diabetes mellitus, diabetes mellitus. 113. Largest gland of the human body is liver, liver, liver. 114. The second largest gland of the human body is thyroid, thyroid, thyroid. 115. Other than kidneys, dash and dash help in the elimination of excretory waste. Lungs, liver and skin. Lungs, liver and skin. Lungs, liver and skin. 116. Sweat glands eliminate nickel, urea, and lactic acid. Nickel, urea, and lactic acid. Nickel, urea, and lactic acid. 117. Sebaceous glands eliminate substances like dash and dash through dash. Sterols, hydrocarbons and waxes through sebum. Sterols, hydrocarbons and waxes through sebum. Sterols, hydrocarbons and waxes through sebum. 118. Secretion of dash provides a protective oily covering for the skin. Sebaceous glands, sebaceous glands, sebaceous glands. 119. Accumulation of urea is called uremia, uremia, uremia. 120. Anticoagulant dash is used when the blood is pumped into a dialyzing unit. Heparin, heparin, heparin. 121. The unit contains a coiled tube made of dash surrounded by a dialyzing fluid. Cellophane, cellophane, cellophane. 122. Antiheparin is also used in hemodialysis. T. When pumping blood back into the body, antiheparin is added. T. When pumping blood back into the body, antiheparin is added. T. When pumping blood back into the body, antiheparin is added. 123. Dash is the ultimate method in the correction of acute renal failures. Kidney transplantation, kidney transplantation, kidney transplantation, kidney transplantation. 124. Renal canaliculi are because of stone or insoluble mass of crystallized salts, oxalates, etc., formed within the kidney. Stone or insoluble mass of crystallized salts, oxalates, etc., formed within the kidney. Stone or insoluble mass of crystallized salts, oxalates, etc., formed within the kidney. 125. Inflammation of glomeruli is called glomerulonephritis, glomerulonephritis, glomerulonephritis.